In this video, I will be discussing the key concepts that are to be observed in our exercise on SDS page. So, SDS page is actually an acronym which stands for sodium dodecyl sulfate polyacrylamide gel electrophoresis. And this electrophoresis is actually a protein analysis. So it's one way of analyzing proteins based on the sizes of the molecules of these proteins. So usually proteins of different sizes are different kinds of proteins, and proteins of similar sizes are similar kinds of proteins. So our sample from which we're going to be getting proteins from to analyze would be human blood. And as you notice here in the illustration, blood is actually composed of two main things. First, we have the serum, or that's the liquid part of the blood, okay, as we see here. And then we have the cells, the solid part of the blood, which are composed mostly of red blood cells or erythrocytes. Now, both in the serum and in the cell portion of the blood, we are actually expecting proteins to be found. So we are also going to take note of both parts in our exercise. So the serum and then the cells. Now the SDS part, the sodium dodecyl sulfate, it's actually a chemical that is a detergent. Okay, it functions as a detergent, meaning to say the molecule of this compound has a portion which is hydrophilic, and there's also a portion, this long tail here, that is hydrophobic. So take note, the proteins in our cells might be mostly embedded in the cell membrane, which is highly amphipathic in nature, and so are these proteins. And so we need a reagent, SDS, which acts like a detergent, which has a portion that is hydrophobic and a portion that's hydrophilic, also amphipathic in nature, so that when the proteins are actually now free from the cell or from the serum, the SDS may actually stick on the proteins, similar to what's illustrated here. So the proteins in their natural state would be highly three-dimensional and coiled in shape, as shown here. Okay, this is in their native state. But once they are exposed to a detergent like SDS, they will actually straighten up like this, and the SDS is going to be sticking on them, okay, and will impart with them a negative charge, as we see here. So they will now be charged negatively. And then the polyacrylamide gel part is, of course, the prefabricated gel that we will be using later on. Okay, it is the medium through which our proteins are going to move through in this electrophoresis process. Okay, so it's made up of a special substance, acrylamide and bisacrylamide. So it's a polymer. So it's a mesh, sort of like your gelatin. And then electrophoresis, of course, is the process by which we use charges to move particles through a medium. In this case, our medium is the polyacrylamide gel, and our particles would be the protein particles. So we have here an illustration of the setup, wherein on the top portion, this is our gel here in the middle, and you can see here sort of there are wells, okay, or hollows on the top portion of our gel where we load our sample of proteins later on. Okay, so using a micropipette. And once the proteins are loaded, we can actually turn on the electrophoresis apparatus. And notice here we have a negatively charged electrode on top and a positively charged electrode at the bottom. And once these are turned on, the positively charged electrode will actually attract now the proteins which have become negatively charged thanks to the SDS that have stuck to the proteins. And so the proteins are expected to travel down the gel, okay, towards the positively charged electrode. And in order to facilitate that, we also add a liquid we call the buffer in the tank, okay, in the electrophoresis apparatus. So this is an illustration of different proteins with SDS already attached to their structure. Now here, letter A is a very short protein, while letter B is a long protein. Okay, and then we put them, for example, in this well, this first well. And here we have a protein of medium length, and say we put it in this well, and then we run the electrophoresis. So notice here, after several tens of minutes, that proteins A and B have traveled through the gel at a different rate. Okay? Protein B is somewhere here on top, and protein A has traveled even farther here. And protein C is just somewhere here in the middle. Now, this has something to do with the sizes of the molecules. Okay? The bigger the protein is, the more difficult it is for the protein to move through the polyacrylamide gel. And the lighter a protein is, like protein A, because it's smaller, the easier for it to move through the polymer polyacrylamide gel. And of course, averagely sized proteins would be somewhere in the middle. So here's a visualization of that. So here we have the different proteins represented by spheres. They are of different sizes. After running the gel electrophoresis, we can see here that the larger proteins have stayed nearer the wells, while the lightest or the smallest of the proteins have moved the farthest away from the wells. Okay, so take note, the gel is actually not solid at all, but then there are spaces in between the molecules of the gel, so the gel is porous. So that's the reason why the proteins are able to pass through it. So we will be loading a volume of 30 microliters of our sample into the wells. Okay, and at the end, hopefully we'll have something like this. So different proteins traveling at different rates. So we can also graph this here against the mass or the size of the protein. So we can see here the heaviest of the proteins are just somewhere here on top, while the lightest of the proteins have traveled really the farthest along the gel. So for our exercise, we'll be loading our protein standard. So that's our standard protein, which will form the bands in well number 1. The serum of our blood sample, the liquid part, will be loaded in well number 3. And the cell lysate, which contains the solid cells and the proteins therein, will be loaded in well number 5. So this is our standard. We call it, the brand is Precision Plus Protein Kaleidoscope. And we will be able to compare later on the result with this particular chart in terms of the sizes of the proteins. So here's a perfectly done electrophoresis. And this is our equipment, this is our tank, and this is our gel. So here it is in the package before it's opened. It's stored in the refrigerator. And here it is after the gel electrophoresis is over. So in order to run the electrophoresis, we need 200 volts for the voltage and 400 amperes for the current, and a duration of 40 minutes. Okay, so we'll also be having a tracking dye so that we can see the colors, the banding colors, and then we will be staining the gel later on after electrophoresis with, again, the Kumase Brilliant Blue, which sticks to the R groups of the proteins in the gel. And then after that, remove the stain, and then we can document our result.